For this lecture, we will cover the Mycobacterium, Chlamydia, Rickettsia, Mycoplasma, and other similar organisms. Mycobacterium are different from the other organisms in the fact that they have an acid-fast cell wall, which requires an acid-fast stain in order to detect these organisms. So that cell wall consists of a very high lipid content, as well as an glycosaminic acid. So these organisms are non-motile, non-spore forming, and they are obligate aerobic. They're very thin to slightly curved or straight rods, and they are intracellular as well. And they are slow growing, but they can take anywhere from 2 to 60 days to grow. And you can see an image here of an acid-fast stain where you actually see those bacilli within that stain. So that would be the pink staining organisms, where the green stuff is debris and cells that are behind those organisms. The Mycobacterium organisms are divided up into two major groups. That is based on their differences in epidemiology and the disease that they're associated with. So we have the Mycobacterium tuberculosis complex, as well as the non-tuberculosis mycobacteria. And then those are further divided into your slow growers and your rapid growers. Mycobacterium tuberculosis complex, these organisms are slow growing and non-pigmented, and they're capable of causing tuberculosis. So this infection could be pulmonary or infected by some other organ. Non-tuberculosis mycobacteria. Mycobacterium leprae is the only organism that is not able to be cultured. These organisms are widely distributed in nature. The infections are acquired through trauma, inhalation, or some sort of ingestion. And these are divided up based on their growth. You have your rapid growers and then your slow growers. And then your slow growers are further divided into the pigment that they produce when they're growing on their special media. So with these, you have your photochromogens. These organisms, you don't have to know this in detail because this is more to do with the testing methods and the organisms that fall within these groups we're not going to discuss. But your photochromers, if they're brought out and exposed to light, they will actually gain a pigment, where your scotochromogens will they'll have a pigment whether they're in light or out of light, so it doesn't matter where you grow them at, they're going to still have a pigment. And then your non-photochromogens, they don't produce any pigment despite where you grow them at. So Mycobacterium organisms can be highly infective because one single organism can lead to severe infection. With your Mycobacterium tuberculosis, it is transmitted person to person with inhalation of the droplets that are contaminated. PPD testing is done to help detect the infection, so you may be familiar with this. You may have had it done or may have to have it done, and this is where they do a test under the skin of your arm, and they look for a reaction to that particular organism. And so a lot of facilities, healthcare facilities, will require new employees to have this testing done, although many have gone to the blood testing method versus the PPD screening test. And then some facilities will require it to be done yearly, although some of them have actually gotten away from actually doing the testing and doing a questionnaire type screening test. Mycobacterium bovis is associated with the ingestion of milk from infected cows. And so this organism will actually penetrate the gastrointestinal mucosal layer and invade the oropharynx. And so there is a vaccine that has been made, a BCG vaccine, that's helped to it's helped to immunize against tuberculosis. However, it's more commonly used in areas that have high incidences of TB. I don't know that it's being used in the United States very much, but there is one available. And of course, individuals who are HIV positive or more susceptible to tuberculosis. So tuberculosis may actually mimic other diseases when it presents clinically, so it can be asymptomatic, but with your symptomatic patients, they will demonstrate pulmonary signs and symptoms, or they can have other organ involvement. Primary tuberculosis is a disease of the respiratory tract where the patient will present with a low-grade fever, night sweats, fatigue, anorexia, weight loss, a productive cough, and those symptoms are a lot of the screening questions in those yearly screening questionnaires. Upon infection, the T cells and macrophages will actually migrate to the lungs, and so those macrophages are attempting to phagocytize the organism, and then the Mycobacterium are capable of intracellular multiplication. And so because of that, the host is unable to eliminate the organism, and then that results in a systemic hypersensitivity to the antigens, and then the granulomas will actually form within the lungs, and then you'll start to have the tissue necrosis that can result from if there's a high antigen concentration of that organism. Primary active tuberculosis, this disease can spread to the lymphatic system, and then this can lead to the meningeal or miliary tuberculosis. It's most often associated with patients with some sort of ineffective cellular immunity. A latent disease is, it has no apparent symptoms or signs. It's not infectious at this point, and it could actually at some point progress to a more active form of it. And so if you do have reactivation of tuberculosis, there's something that has caused this activation, so some sort of incident with the cellular immunity that's been suppressed or damaged that causes that reactivation. And of course, again, individuals with HIV or more susceptible to developing an active tuberculosis infection. So there can be other organ involvement with a tuberculosis infection. So you can have the genitourinary tract, lymph nodes, the central nervous system, bones, joints, the pleural lining of the lungs, and of course, again, that TB test that I mentioned earlier, where they put that little bubble under the skin. And then you also have the quantiferon TB Gold, which a lot of places are using that method now, where they take the blood sample and send it off for the testing. Your non-tuberculosis Mycobacterium are divided into slow growers and rapid growers. You also have the Mycobacterium leprosy, that is not culturable. But our slow growers can also be divided further into photochromogens, scotochromogens, and non-photochromogens. So the Runyon group of classifications has been used. So your Runyon group number 1 is your photochromogens. And with these organisms, the colonies, when growing on the agar, will develop a pigment after they've had exposure to light. So you would grow them in the dark, and then once you look at that organism, you would expose it to light, or you could grow the organism in the dark in a separate culture in the light. and you're looking for that color change due to the exposure to the light. And so with these organisms, they are slow growers, so it will take them more than 7 days to actually appear on the solid media. The scotochromogens are organisms that will develop the pigment whether they're in the dark or in the light, and these are also slow growers, so it takes them longer than 7 days. The non-photochromogens, they will not develop any pigment regardless of whether they're grown in the light or dark, and they also take longer than 7 days to appear on solid media. And your rapid growers will grow less than 7 days when they are growing on solid media. Now keep in mind that there are many Mycobacterium outside of the ones that we cover, but we're going to cover the ones that are most likely seen in some sort of clinical specimen or infection. And so with Mycobacterium canzii, this is in group 1. And so these infections typically are found in natural reservoir or tap water, and they can also spread via aerosols. With these organisms, they do cause chronic pulmonary diseases, extrapulmonary diseases such as cervical lymphadenitis and cutaneous diseases. And with the Mycobacterium mammarium, this organism can be found in freshwater and saltwater as a result of contamination from infected fish or marine life. And so these organisms are transmitted through contact with the contaminated water, and the organism will usually enter with some sort of, due to some sort of trauma or break in the skin. And so it is associated with aquatic activity where fish are present. And this one does cause a cutaneous type disease of bacteremia. Our group 2, again, these are the organisms that do have a pigment when they're grown in the dark or the light, regardless. They're rarely recovered. However, there are a couple that can be seen. And so Mycobacterium scophosrium, you can find this within raw milk, soil, water, various dairy products. And it tends to cause cervical adenitis in children, bacteremia, pulmonary diseases, skin infections. And with the Mycobacterium gordoniae, this one can be found in tap water, any water source, soil. It's typically non-pathogenic. Rarely will it ever cause an infection, but you can sometimes see it as some sort of contaminant in some specimens. Within group 3, these are the organisms that do not produce any pigment, and these are pathogenic. So within the Mycobacterium avium complex, you do have several sources, which includes natural waters and soil. And it's usually within patients that do not have AIDS. It can cause pulmonary infections. For those patients that have some sort of pre-existing pulmonary disease, as well as it can cause disseminated type infections, diseases within these patients. Within your patients that are immunocompromised who do have AIDS, it can also cause disseminated infections within those patients. Of your non-tuberculosis Mycobacterium, your most commonly isolated would be your Mycobacterium avium complex. And so this complex actually consists mainly of the Mycobacterium avium and Mycobacterium intracellular. And so these organisms cause infections of the respiratory tract in adults and the lymphatic system in children, and it can cause disseminated infections in patients who have HIV. And the incidence is higher in patients who are immunosuppressed for any reason, so it could be something like chemotherapy. Identification of this organism is based on the fact that they are slow growing and they're non-photochromogenic, so they have no pigment when they're growing on the special agar. And so you can see an example of that here, where they just kind of have a buff look to them. And of course that pigment doesn't matter. They have no pigment regardless if they're grown in the light or in the dark. Group 4 is our rapid growers, so you will see growth within 7 days. And these organisms can actually grow on routine media, and they can appear as weakly gram-positive on a gram stain. But most commonly these infections are due to some sort of post-traumatic wound type infection. And so Mycobacterium fortunum is found in different environmental resources, including just natural water supplies or municipal water supplies found within the soil, and you can get these as healthcare associated infections as well. And so you can see these types of infections in post-operative infections, like with a breast augmentation. You can find them in skin and soft tissue infections, pulmonary infections, and different localized lesions, which can be rare, but if they are found, they usually do have within the central nervous system a very high morbidity and mortality rate. Mycobacterium leprae is transmitted person to person through inhalation or direct contact with someone who has an infection or infectious skin. Infections are rare in the U.S., but there are still some cases that are seen in Texas and Louisiana. And so this particular organism causes what we know as leprosy or Hansen disease. And so this is a chronic disease of the skin, mucous membranes, and then it can affect the nervous system. There is a silent phase in which the bacilli will multiply within the cell macrophages. And then there's an intermediate phase, which they will actually move on to the peripheral nerves and cause sensory impairment. So there are two forms: a tuberculoid, which is there's no immune deficit, so there's a localized just to the skin and the nerves, and then you have very few organisms that are actually present in those lesions. And then you have the more common form, which we associate with leprosy, which causes an extensive skin lesions, and then you have the numerous acid-fast bacilli within those cells. And with this organism, it cannot be cultivated, so you cannot actually grow this one in the lab. Or rather, you cannot grow it on culture media. So with this organism, it's going to be identified based on your direct detection of the skin cells. So you're looking for doing that acid-fast test, and you're actually looking for those organisms here up top. You're looking for them, they're the pink ones, inside of those skin cells. This green stuff is just debris or skin cells of some sort. And so you're, with this particular technique where you're looking at that acid-fast to be positive, it helps to confirm that that is what that infection is. Some of the other methods that are used would include an actual cultivation within the foot pads of mice. And so that's what you see here in this top image is a mice that have been inoculated with this organism through some sort of culture technique where they got some sort of cells or something from the patient and inoculated that mouse foot. And so you can see the swelling that happens from those macrophages, and you can also see an example of a patient here where those macrophages have caused destruction within that skin. And then the other culture method would be to culture it in a 9-banded armadillo. So you see your armadillo here. Why 9-banded? I don't know. I guess 8's not enough, but that is one of the other methods of which this organism could be cultivated. So to detect these organisms, it's best done by using acid-fast staining. So there are a couple of different types of technique. You see an example of a fluorochrome acid-fast stain up top, and then you have the Kenyon stain at the bottom where you actually see those pink rods. And then of course you have automation where you're looking for the different antigen proteins, genetic sequencing, nucleic acid testing, DNA type testing. And if you're doing conventional methods by growing it in the lab, you're trying to determine the growth rate of the organism to help classify it. So when you're growing these organisms, once you start seeing growth, so if it's day 3 and you see growth, then it's considered a fast grower, which narrows it down. And then if you get past day 7, then those are slow growers, so that kind of narrows it down. You're looking at the pigment production of the organism, and then there's also biochemical testing that can be performed. So here's a chart just to compare these different organisms here. And again, the Mycobacterium leprae does not culture, so it is not included with the culture characteristics. And here at the bottom, your tuberculosis organism growing, the colony morphology on the special agar does demonstrate like a roughness to it, whereas your other organisms will appear more smooth. So image A is your Mycobacterium tuberculosis, and B would be an example of the others. Next, we'll cover our obligate intracellular and non-culturable bacterial agents. So with your obligate intracellular bacteria, they're usually very difficult to cultivate, or you're unable to cultivate them at all. And so this includes your organisms like your Chlamydia or your Rickettsia. And so these differ from most of your other bacteria because they're very small and they're obligated intracellular parasites. So they actually need a cell to live inside of. Bartonella were previously classified under the Rickettsia, the Rickettsia type organisms. However, over the years there's been some differences that have been found within their genetic makeup, so they have been classified into their own distinct order. Your chlamydia organisms are obligate intracellular bacteria, so they do look very similar on a gram stain to a gram-negative bacteria, and they do contain a lipopolysaccharide in the cell wall, but they also have a major outer membrane protein. These organisms do not exhibit much of an endotoxic activity. Some of the infections that they cause are STDs, eye infections, and respiratory infections. So these organisms do have a very unique life cycle. So again, because they are intracellular, they do need a cell to live and thrive in. And so what you will have is an actual elementary body that will attach to the cell, the host cell. And so like this could be like an epithelial cell. And then from there, once it attaches to the host cell, it will actually be engulfed by that host cell. And then from there, those elementary bodies will reorganize and make a reticulate body. So you see that large reticulate body here in the center of this. And then from there, they can multiply by binary fission. And then within that host cell, and then those inclusions will actually turn back into those elementary bodies, and then they will lyse that cell, and then they're released back. And so that continues their life cycle to start over again. Here we have a chart that compares the different organisms based on their general characteristics. And so you can see that Chlamydia trachomatis is an infection of humans, whereas your Chlamydia pseudococci affects birds. It's carried in birds, and then it can cause infection within humans. And then Chlamydia pneumoniae causes the infection in humans. And your Chlamydia pneumoniae, with its elementary bodies, will actually take a pear shape, whereas the other two are round. Chlamydia trachomatis is transmitted by human-to-human direct contact of any sort of infected secretions, and then it can also be passed from the mother to the infant during birth. Infections are often asymptomatic, resulting in transmission, so if the patient doesn't know that they have this infection, then obviously they could transmit it without knowing. And so this is the most common bacterial STD pathogen. It is associated with infertility, ectopic pregnancy, as well as pelvic inflammatory disease. And it can also cause an ocular trachoma. Chlamydia trachomatis has different serovars that cause different clinical syndromes, and so those are divided out here. And it can range anywhere from eye infections to genital tract infections, respiratory infections, lymph node type infections, and they're all transmitted by different methods. Trachoma is a chronic infection of the conjunctiva, and so this is a very preventable disease, but it has been known to cause blindness worldwide, and it causes irritation of the eyes and eyelids, and can produce a lot of discharge. As the infection increases, it causes eye pain, blurred vision, photophobia, and then repeated infections result in scarring of the inner eyelid. So you can actually see an example here at the bottom where you have some of that scarring of that eyelid. Lymphogranuloma is a sexually transmitted disease, and it appears with genital lesions. So these lesions will actually appear within the lymph nodes around the genital area, and it can progress to a third stage, causing the development of genital hyperplasia, fistulas, and different other problems and complications. Oculogenital infections infect both adults and newborns as a result of genital secretions that come in contact with the eyes. And so some of the other ways that it can be transmitted would include swimming pools or hot tubs that have not been properly chlorinated, or through sharing makeup. So this organism can be cultivated through McCoy agar, HELA agar, monkey kidney cells, or it can also be detected through direct detection of the antigens or the nucleic acid amplification testing and different serological type testing methods. Chlamydia pseudococci is a pathogen of birds, so it's a variety of birds that it does affect, but it is most associated with parrots, and therefore it is named parrot fever. And so parrot fever is not Jimmy Buffett fans that are looking for 5 o'clock somewhere, but rather an infection in which the patient will actually inhale the bacteria in aerosols, and then they will present with a pneumonia type infection with severe headaches, mental changes, hepatosplenomegaly. So this organism is diagnosed by serological type methods, as well as it can be diagnosed by PCR testing. And this organism is resistant to the sulfonamides. And then the elementary bodies and inclusions with this particular organism differ a little bit from the trachomitis. Chlamydia pneumoniae It spreads person to person via aerosols and causes a mild to moderate respiratory infection. And so this would be something like pneumonia, bronchitis, sinusitis, or flu-like illness. It infects many cells that are associated with the respiratory and cardiovascular tract. And so what it will do will actually infect those cells, like your epithelial cells or your alveolar macrophages or your circulating monocytes. And then when it infects those cells, they will actually cause if you were to visualize those cells under a microscope, then it would actually cause a pear shape to those elementary bodies. So here are those elementary bodies that have taken on that pear shape. And to diagnose this, you could use a cell culture, serological testing, or NAT testing, the nucleic acid amplification technique. Rickettsia is transmitted by arthropod vectors. So this will be something like ticks, lice, fleas, chiggers. This causes a spotted fever group or typhus group of infections. The one that you may be most familiar with would be Rickettsia rickettsii, which causes Rocky Mountain spotted fever. And so you can see an image here with someone who has been infected with this, and they've got those spots after that infection. And so this is usually diagnosed through different direct detection methods, immunohistology, PCR testing, serodiagnostics, and again, just to remind you that these organisms do not grow well in culture, so that's why automated testing is necessary for them. Anaplasma of Phagocytophilium. This organism is transmitted by ticks, and it infects the bone marrow derived cells. And so those would be like the cells that come from the bone marrow, red blood cells, white blood cells. And particularly, it does primarily infect the neutrophils. And the infection that it causes is human granulocytic anaplasmosis, or HGA. And what you have is the organism actually goes into the neutrophil. So this is a neutrophil. The granulocytic is because neutrophils do have granulocytes. So you can see all of those little granules within this neutrophil. And then this right here is the actual organism that is invaded that cell. It causes a mild illness that can be mistaken for viral infections. It does take a very trained eye to notice the organism inside of the white blood cells. If they were to do a differential and actually look at the white blood cells on a slide, you can see that it looks very similar to these granules, so the technician would have to actually be able to recognize that. So you can detect it through an actual stain, but as you can see, with it being a little bit more difficult to recognize, that your antigen antibody testing, immunofluorescent antibody testing would be the best way to diagnose this organism. Rickettsia chafensis is transmitted by a tick, specifically the Lone Star tick. It primarily infects monocytes, and so just like it infects the neutrophils or the granulocytes in the previous organism, this one infects the monocytes. Therefore, the infection is termed human monocytic ehrlichiosis. And you can see here where this is the monocytes. And monocytes do not have granules, but you can see the organism here inside of that monocyte. So it causes non-specific symptoms like fever, headache, tired, weak, rashes rarely occur. It is identified based on its appearance on a peripheral blood smear stain. It can also be detected through immunohistology, PCR, or serodiagnostic type testing. Coxiella burnetii. So this organism is a zoonotic type disease, so it is transmitted from animals and it is inhaled. And this is known as the causative agent of Q fever. And so it does cause an acute infection of the lungs. These organisms are small gram-negative coccobacilli, and these organisms are a little bit different because they can actually survive extracellular, so they can survive outside of an actual cell, whereas most of the others, they need to be inside that cell. So cultivation can take place using human lung fibroblasts, but this is considered a biosafety level 3, so this particular type of testing would not be performed. Serological testing would be most likely what would be used for identifying this organism, as well as PCR testing. So your cell wall deficient bacteria include your Mycoplasma and your Ureaplasma. And so these organisms are transmitted through direct sexual contact, transplants, mother to fetus, or through respiratory secretions. They are highly fastidious, slow-growing, facultative anaerobes, and they are very small in size. They have no cell wall, so they will not gram stain. So if you were looking for this organism under a gram stain slide, you would not see it. These organisms colonize the mucosal surfaces of the respiratory and urogenital tracts, and they do this by attaching with great affinity to the surface of the ciliated and non-ciliated epithelial cells, and they cause a long-term inflammatory syndromes and can cause cell death. Some of our important species that would be isolated would include the Mycoplasma pneumonia, which has a P1 adhesion protein, your Mycoplasma genitalium, which has a MGPA adhesion, Mycoplasma pneumonius, and Ureaplasma urolithium. Mycoplasma pneumoniae can cause infections that are anywhere from asymptomatic to a little bit more severe with upper and lower respiratory tract infections. And so this causes community-acquired atypical pneumonia, which we also know as walking pneumonia. And it's because this organism can attach strongly to the mucosal surface cells. Ureaplasma urolithium and Mycoplasma pneumonius can cause systemic infections in neonatals, as well as invasive disease in patients who are immunocompromised, as well as urogenital tract infections. Mycoplasma genitalium is a non-gonococcal uritis in men, and as well as it causes cervicitis and pelvic inflammatory disease in women. And you can see an image here where you have that infection of the cervix. These organisms are diagnosed based on PCR or serological testing methods. If they are cultured, they're best cultured using the beef or soybean protein with serum, with fresh yeast extract, and other specific growth factors. And with these organisms, when they're plated on this particular media, the colony is very hard to see, so you actually have to take the agar plates and look at it in some sort of microscope to actually see the appearance of the colony on that agar. And so when you do that, you're actually looking at these two images here to the right. So for like the Mycoplasma pneumoniae, it would be a spherical shape that's kind of grainy. And then with like your ureaplasm, it can be a darker brownish color, so you can see that there at the bottom. And then your Mycoplasma pneumonius would have like a fried egg type appearance. So you can see that here up top where the arrow is pointing to A, where you have those large culture cells that kind of have that fried egg type appearance to it. So these organisms are slow growers, and so with these slow growers, and the particular type of culture method that has to be used to identify them, your PCR and automated methods would be the better option. Nocardia organisms are typically found in soil, and they're usually transmitted due to some sort of penetration to the skin or abrasion to the skin. And they can also be inhaled as a mode of transmission. With Nocardia asteroides, it is a facultative intracellular pathogen that's able to grow in various human cells. So this is one of its virulence factors that allows it to be a pathogenic type organism. These organisms also have different stages of growth at the time of causing the infection, and they are resistant to being killed intracellularly. And they can also inhibit any sort of phagosome-lysome fusion, and they also can produce catalase and homolysins that prevent them from, that aid in their infectious state. So there are typically two organisms that will cause infection. With Nocardia asteroides, this will cause a pulmonary infection in immunocompromised, whereas Nocardia brasiliensis will cause cutaneous infections in patients who are immunocompetent. In other words, they have a healthy immune system. And with a cutaneous infection, it can turn into a mycetoma, which will cause a chronic, localized, painless subcutaneous type infection, as well as it can cause some other infections with the lymphocutaneous infections and skin abrasion, cellulitis type infections. The microscopic and colony morphology can resemble a fungi, so you can sometimes see this organism included in fungi lectures or mycology lectures, but it is a bacteria. And so if you were to gram stain it, it can appear as a gram-positive organism with a beaded type appearance where it's branching, or it has very fine, delicate filamentous or fragmented type appearance. So they may not gram stain, so you may potentially need to do a modified acid-fast or acid-fast stain with these. And so as the organism ages, it will actually start to get more of that fungi type look to it, where it will start having fragments or start with the pleomorphic rods or coccoid elements. And so you can see that beading type appearance in the stain as it ages. Your Nocardia organisms can have a variable appearance when they are cultivated and they're growing on the agar. And so if they were to grow on a blood agar, then it could actually produce a beta hemolysis. But they typically look dry or a chalky white appearance to an orange tan pigment, and they can kind of have a crumbly look. And as they age a little bit, they can start to produce those aryl hyphae, which can make it have an appearance similar to some of the fungi. If you remember from our yeast lecture, some of the organisms would do that wrinkly type appearance. So these organisms can grow on non-selected media, but it can take them longer, 3 to 6 days. So unless you're trying to suspect this organism and you want to see if it grows on the blood agar, typically these cultures are going to be thrown out before you'd actually see it growing. And there are a couple of different types of media that can be used to help recover this organism. They are catalase positive, nitrate positive, urea positive, and lysozyme resistant positive.